Assalamualaikum warahmatullahi ta'ala wabarakatuh. Welcome to our channel. This time around, we will be presenting on the epigenetics of breast cancer, which is a part of molecular cancer research. This paper concerns the modifying role of environmental and bioactive food compounds, which is a review article published in June 2016. The abstract of this paper consists of scope, followed by methods and results, as well as conclusion. The scope of this assignment revolves around the expression of tumor suppressor genes, TSG, endocrine disruptors in foods and drinking water, as well as the hypothesis of this experiment itself, which is the agonist of AHR, BPA, and arsenic compounds induced in TSG. The methods and results of the experiment is rather straightforward, as it revolves around the overlaps between epigenetic signages with epigenetic alterations. This is followed by the opportunities to prevent sporadic breast cancer with food components. In conclusion, this review article characterizes the overlap, which gives new prevention strategies with specific food components or diet. This review article aims to develop a working hypothesis that endocrine disruptors induced in tumor suppressor genes, TSG, epigenetic signages that mirror those seen in sporadic breast tumors. It starts with the introduction about sporadic breast tumors, which represent most breast cancer cases, the affected individuals, which are the same person, as well as the generations after, the definition and concept of epigenetics itself, which consists of the changes in gene expression, as well as the absence of changes in DNA sequence. The researchers made use of 5 steps or procedures to complete this review article. Initially, they carried out systematic review, which involves PubMed literatures, followed by the consultation of TSGene 2.0 database, PubMed searches, as well as other searches involving other databases, and finally, comparison of studies.
Next are the results. The first part of the results is regarding the mechanisms of epigenetic disruption of tumor suppressor genes in sporadic breast cancer. Tumor suppressor proteins. microRNA and long non-coding RNA.
So, for prevention of epigenetic disruption with food component, the epigenetic factors include AHR agonists, BPA, and arsenic. For food that can combat AHR agonists include resveratrol, genistein, cruciferous indoles, and other food components such as curcumin and caffeic acid. For resveratrol, example of studies include in cell culture experiment with MCF10A cells, which is a type of epithelial cells. Resveratrol protected against PAH-induced DNA damage, which is PAH is a polycyclic aromatic hydrocarbon, and resveratrol protected against damage done by PAH, and it also suppressed expression of CYP1A1 and CYP1B1, which is a type of biomarkers for hypoxia damage. So that's what resveratrol do. For genistein, example of studies include in Sprague-Dawley rats, the perinatal exposure from conception through day 21 to postpartum was correlated with enhanced mammary gland differentiation and reduced DMBA-induced mammary tumorigenesis. DMBA in this context means immunosuppressor or a type of carcinogen, and genistein reduce the DMBA-induced mammary tumorigenesis. The preventive effect of genistein against breast cancer have been related to inhibition of DNMT1, leading to demethylation in tumor, tumor suppressor gene. Next, for cruciferous indoles, for example, sulforaphane, which is an isothiocyanate found in cruciferous vegetables. This food have studies done on it, and it shows that SFN protect against the MBA-induced mammary tumorigenesis. Next, other food component that can handles the effect of AHR agonists in breast cancer include curcumin, and the effect of curcumin were confirmed in hepatocellular carcinoma cell, in which it inhibits phthalate-dependent expression and activation of AHR, migration, invasion, and EMT. Another food that can combat AHR include caffeic acid, which was shown to demethylate MR148 and abrogate cancer stem cell properties. Next, for BPA, food that can prevent the epigenetic disruption include genistein. A study, the maternal supplementation with genistein at level consumed by population on high soy diet, was shown to modify coat color of heterogeneous viable yellow agouti rat. This phenotypic change was correlated to increased methylation of CpG sites. Another food component that can combat BPA include naringenin, citrusflavone, curcumin, and all of these showed its effect of antagonizing BPA-induced proliferation. For arsenic epigenetic disruption, folate and vitamin B12 have been shown to prevent the effect of arsenic in breast cancer. For folate, it regulates the pool of available metal groups, which can be effective in lowering the risk of breast cancer. For vitamin B12, intake of B vitamins may influence the epigenetic response to arsenic. The combined action of folate and B12 may protect the one-carbon metabolic pathway and reduce arsenic-induced mutagenic DNA breaks and tissue damage. So that is all for prevention of epigenetic. So, in conclusion, because most breast cancer cases are sporadic, discovering the epigenetic mechanism that regulate tumor development may offer new target for prevention and treatment. This discovery of epigenetic mechanism can allow us to find a way to develop a treatment that can target the epigenetic mechanism itself. Throughout the history, environmental pollutants, foods, and drinking water are sources of xenobiotics, including agonists of the AHR, PAH, dioxin, phthalates, PCB, BPA, and arsenic, which may contribute epigenetically to dysregulation of tumor suppressor gene and breast cancer. Overall, studying the epigenetic mechanism in breast cancer and finding out dietary compounds that can prevent this epigenetic factor can be a potent prevention in breast cancer and any other kind of cancer.